Hi students, I am A. Monica, working as an assistant professor in the Department of Genetics and Plant Breeding in Best Innovation University. Today I am going to discuss about crop improvement in rice, and it includes taxonomical classification, breeding objectives, breeding methods, and what are the breeding methods involved in hybrid rice production. First we will discuss about taxonomical classification. The scientific name of rice is Oryza sativa, and kingdom, it belongs to the kingdom Plantae, and order Poales, and family Poaceae. Next, genus was Oryza, species was sativa. And the crop was originated in Southeast Asia, and chromosome number of rice is 24, and it got distributed in mostly humid tropical and subtropical climate, and 90% of production and consumption happens in Southeast Asian countries. Major producing countries are China, and it was followed by India, Japan, after that Korea, Pakistan, Bangladesh, and other Southeast Asian countries. And in India, the major producing states are West Bengal, and it was followed by Uttar Pradesh, after that Andhra Pradesh, Karnataka, Tamil Nadu, and Orissa. The genus Oryza having 23 species. Among those 23 species, 21 are wild species and 2 are cultivated species. And this genus having both diploid and tetraploid species, and it includes the genomes like A, B, C, D, E, and F. The two cultivated species of rice are Oryza sativa, the common name is Asian rice, and next one is Oryza glaberrima, that is African rice. We will discuss about origin of cultivated rice. What are the cultivated rice? That Asian rice and African rice. When coming to the origin of cultivated rice, it is having two classes. Those are polyphyletic origin and monophyletic origin. And coming to the polyphyletic origin, this Asian rice and African rice, that Oryza sativa and glaberrima, it has independently evolved from several subspecies having one common ancestor, that is Oryza preperennis. And here we will see the classification. Common ancestor is Oryza preperennis, and coming to the Southeast Asia and tropical Africa, it was independently evolved. Coming to Southeast Asia, the Oryza rufipogon, which was wild species, from that Oryza nivarva get evolved. After that, Oryza sativa and Oryza spontanea get evolved. From Oryza sativa, it is having three main ecotypes, and what are those? Indica, Japonica, and Javanica. And coming to the tropical African region, the Oryza longistaminata get evolved. After that, Oryza barthi. From Oryza barthi, both Oryza staphy and glaberrima got evolved. Here I have given the classification of Indica, Japonica, and Javanica. Here we will discuss about distribution. It got distributed in tropical regions, that is India, southern China, Vietnam, Thailand, Myanmar, Philippines, Bangladesh, and Sri Lanka. Next, when coming to the Japonica, it got distributed in subtropical climate and warm temperature regions. Those includes Korea, Japan, and northern China. And next, Javanica got distributed in subtropical climate, or you can say it as an equatorial climate, that is Indonesia. And coming to the tillering capacity, it is having profuse tillering, and it is this Japonica having medium tillering capacity. And coming to the Javanica, it is having low tillering capacity. And coming to the height, it is tall, next one is short, after that tall. And grains are here little bit slender flat grains, and here short and roundish grains. There broad, thick, and somewhat coarse type of grains. And coming to the temperature, this Indica variety having sensitiveness, and these two are Japonica and Javanica having resistant characters. And coming to the drought conditions, this one is resistant variety, and these are intermediate, and those are sensitive varieties. And next, yield potential, here they are giving medium yield, and here when we will come to Japonica, this is giving high yield potential, and last one is low yielding potential. And coming to the monophyletic origin, in this the two cultivated species, that is Asian rice and African rice, both got evolved from one common ancestor, that is Oryza perennis. Here the common parent is Oryza perennis. From those Oryza perennis, Oryza sativa and glaberrima got evolved. Here I have included some of the wild species. First one is Oryza nivara. Here we can see the picture of the these wild species, Oryza nivara, and it is resistant to grassy stunt virus and bacterial blight. Grassy stunt viruses, it is transmitted by green leaf hoppers. And Next one is Oryza rufipogon. This is also one of the wild species, and you can see the picture of this crop. This is resistant to bacterial blight, and this is resistant for pests like brown plant hopper, that is Neelaparvata lugens, and it is having resistant character against Tungro virus also, and this is the source of CMS, that is cytoplasmic male sterility. Here we can introduce cytoplasmic male sterility from wild species to cultivated species. Next one is Oryza burthi, and you can see the image of this wild species, and it is resistant to green leaf hopper, that is Nephotetics viruses, and it is resistant for bacterial blight also, and it is drought avoidance character, and it is tolerant for heat. Next we will discuss about Oryza longistaminata, one of the major wild species, and it is resistant for bacterial blight, and particular character is resistant for nematodes, and it is resistant for stem borer, and it having drought avoidance capacity. So next we will see floral biology. Here we can see the bottom stalk-like portion, that is the pedicel, and two rudimentary glooms are there that are not fully developed, and sterile lemma will be present that are also rudimentary, and those are in sterile nature. And coming to the outer portion, that is lemma and palea, and inside that female and male reproductive organs will be present. Male reproductive organs consists of two parts, anther and filament. This stalk-like structure is called filament, and top one is anther portion. This together consist of stamen. And coming to the female reproductive organ, these consist of ovary, style and stigma. The stigma is in bifid feathery nature. And here where that on portion will arise, that portion is used to call apiculi, and this spike-like structure is called on. In most of the wild species you can observe this portion, on portion. And coming to the next, here the opening of the spikelets, happens from top and it proceeds towards downwards, and here the complete opening of the total florets, it will takes place up to 4 to 7 days, nearly 1 week, and the anthesis, the blooming of the spikelets happens at 10 am to 2 pm. The lemma and palea will closes after the shedding of the pollen grains, and overall the maturity of the grains will take place around 30 to 35 days. And next, after maturation of lemma and palea, it turns into the husk portion. That fruit used to call as caryopsis. Next we will see the breeding objectives. So in those breeding objectives, first we are going for yield potential, to increase the yield. Next one, to increase the adaptability for suitable environments, and yield should be stable according to the environments. And you need high early maturing, medium maturing and long maturing varieties. That means based on the duration we have to develop the varieties by following certain breeding methods. And next one, the crop should be non-lodging type and non-shuttering type, so that we used to call resistant to lodging and shuttering nature. And next, it should be resistant for cold temperatures. Next, resistant to salinity and alkaline natures, alkaline soils. So it should show resistance against saline soils and alkaline soils, and resistant to diseases like blast. This is the major disease in the rice. Next one, stem rot, Tungro virus and sheath blight. And next, it should be resistant for pests like stem borer, gall mites and brown plant hopper. Next, to improve the grain quality. After that, it should be act as a alternate source for dwarfing gene, and it is suitable for direct seeding, that is broadcasting. And these breeding varieties should be suitable for the dryland conditions and also for deep water conditions where the water will be stagnant. And these varieties should be suitable for exported, and it should be scented varieties. Those are basmati varieties. And breeding varieties should also control the wild rice, and these varieties should be suitable for any unsuitable conditions also. And next, coming to the breeding methods, first one is plant introduction. That means we are introducing the crop variety from different countries or different states to our native countries or our native states. So from IRRI, that is International Rice Research Institute, from those institutes we have introduced the varieties like IR8, from IR8 to IR72 varieties. And other examples I included was Basmati from Punjab and Ponni Masuri from Malaysia. And next one is CR1009, that is Ponmani from Orissa. Next one is pure line selection. After that, hybridization and selection procedure. And in among the hybridization, intervarietal hybridization, that we are crossing two different varieties, and between two races, that we used to call interracial. And for example, I have given Pony Masuri, the parentage is Taichung 65, that we are crossing with the male parent, that is ME 80, and interspecific crosses, that we are crossing between two different species. The developed variety was Coimbatore 31. The parentage is Oryza perennis, that we are crossing with the male parent GEB 24. It is resistant for the drought. And next breeding method is back crossing method, that we are crossing F1 hybrid with any of its parents. Next breeding method is mutation breeding. So first induced mutations was created by Ichijima in the year 1934 by using X-rays and UV rays. And next in 1938, this mutation research was totally initiated in India by Ramaya and Parthasarathi. Next, this chemical mutagenesis in rice was introduced by Sukanya in the year 1957. In 1962 to 1963, Mary, she used some chemical mutagens. That mutagens are diethyl sulfonate and ethyl methane sulfonate. And in 1964, FAO, that is Food and Agriculture Organization, they initiated some coordinated program on rice breeding with induced mutations. So next one is heterosis breeding. Heterosis means that F1 hybrid is performing better than its parents, or you can say it as a superior performance of F1 hybrid than its parents. So this is the universal phenomena, because F1 progeny is showing better performance than its parents. So varieties developed was KRH1 and KRH2. This parentage is IR 58025A. We are crossing with the IR 9761 10 IR. This is the parentage of KRH1, and KRH2, the parentage is IR 58025A and with KMR3. Next, coming to the hybrid rice breeding technology. This involves mainly three breeding methods. Those are 3-line system, 2-line system and 1-line system. So first we will discuss about CGMS system, that is 3-line system. So see here, male sterile length, that you can use it as a female parent in the hybrid seed production, that we are crossing with the male parent, that is male fertile line, that we used to call male line or maintainer line. Why you are calling it as a maintainer line? Because it used to maintain the male sterility in the female parent, that is A line. If you are crossing A with B line, then you are getting hybrid as a male sterile line, that is A line, or you can say it as a male sterile line. If you want to restore the fertility nature in the hybrids, so we have to cross A line with the male restorer line, that is R line, or you can say it as a restorer line also. When we are crossing with A line with R line, we are getting the fertile hybrid, that is male fertile line. So this overall system is called 3-line system, or you can say it as a CGMS system. Next method is 2-line method. In this 2-line method, it includes mostly three ways. So first one is mechanical means, by mechanical emasculation and all these things, and next one application of male gametocytes, and next by inducing the environmental genetic male sterility. Among these things, so it is mostly classified into 3 types: PGMS, TGMS and PGTMS. What is that? PGMS is nothing but photosensitive genetic male sterility, and TGMS is thermosensitive genetic male sterility, which has been successfully used in China. And next, in PGMS, the photoperiod length should be at least 14 hours. If it is more than 14 hours, it will result the hybrids as steriles. And next, if it is less than 13.45 hours, it is partially sterile hybrid. If you want to get fertile nature hybrid, we have to maintain photoperiod length up to 14 hours. That is the maximum limit of photoperiod length. And next, what is the first PGMS line they developed? Nonggen 58S. This is the first PGMS line, and it was discovered in the Japonica variety. Next, TGMS line, thermosensitive genetic male sterile lines. So if the day temperature is 31 degrees centigrade and night temperature is 24 degrees centigrade, this will result in the sterile hybrid. and if it is temperature is 28 degrees centigrade and night temperature is 21, it used to results in partial fertile hybrid. And next, if the day temperature is 25 degrees centigrade and night temperature is 18 degrees centigrade, it will result in fully fertile hybrid. And first TGMS line developed was Anong 1S, and here we are discussing critical sterility point and critical fertility point. Critical sterility point is nothing but the temperature we are maintaining to induce the sterility nature. And what is critical fertility point? The temperature we are maintaining maximum to induce the fertility. So next method is one-line method. It includes mostly asexual propagation. Among those, vegetative propagation, micropropagation, anther culture, it results in the haploids and apomictic lines. So this completes the breeding methods for development of the hybrid rice. Rice crop improvement was completed. We will meet in the next class. Thank you.